This is an update on the progress on our grant for the past year, focused on the preclinical development of a ULK1 inhibitor for pancreatic cancer. So just to remind everybody, the focus is on the inhibition of autophagy, which is a cell survival mechanism that is highly upregulated in advanced cancers. Specifically, we're targeting the upstream serine threonine kinases, ULK1 and ULK2, which are autophagy initiators. And so the inhibition of these target kinases leads to the inhibition of autophagy and treatment for cancer. Just so over the last few years, there has been significant progress on the validation of autophagy inhibition as an approach to treat highly advanced cancers, and especially pancreatic cancer. The mechanisms around this are shown on this next slide. So with respect to the tumor microenvironment, as I mentioned, autophagy is highly upregulated in advanced cancers. And this leads to the accumulation of tumor cells, especially in the hypoxic tumor core of solid tumors such as pancreatic cancer. Unfortunately, all cancer treatments, so that's standard chemotherapeutics, newer targeted therapies, and also radiation therapy, lead to the upregulation of autophagy. And the proposal is that this is a reason for the resistance in cancers following treatment. Now, so far, the tool compounds and therapeutics that inhibit autophagy have not really been available. The only compounds which have been used clinically are hydroxychloroquine and chloroquine, which are very old anti-malarial drugs. And these compounds are not very potent, and they also have many off-target effects, pleiotropic effects specifically. Work from the McMahon lab and others have shown that the inhibition of autophagy in pancreatic RAS-driven cancers, combined with the inhibition of the RAF-MEK-ERK pathway using drugs such as trametinib, is highly effective at stopping pancreatic cancer. Now, back in 2015, we published a chemical probe, which was an early ULK1 inhibitor. This is a selective compound and a useful cellular probe that demonstrates effective inhibition of autophagy in cells. So the purpose of this studies are defined in specific aims of the grant, and that is to advance a compound ultimately into the clinic to treat pancreatic cancer. And this has involved the scale-up of our lead candidate compound, continued biological studies around the lead candidate and other compounds in the series, and also the performing initial IND-enabling studies around the lead candidate. Now, we were very pleased to receive a third year of funding from PanCAN, and because of this, we added a fourth aim, which was to perform studies to identify a backup ULK1 inhibitor to treat pancreatic cancer. So going through the specifics of our progress on this, so this slide relates to aim 1, and the summary here shows our success in being able to improve the process of the synthesis of our lead candidate to the point where we have now produced over 100-gram batch of the compound. And this is critically important in terms of the amounts of material we will need to do non-GLP IND-enabling studies. So this is hot off the press, but again, our latest batch was produced from 92 grams of the key intermediate, producing more than 100 grams of the lead candidate. With respect to AIM2, and so these, the in vivo studies are performed by my collaborator, Professor Ruben Shaw at the Salk Institute. And this is just a reminder that his lab was able to take our drug, SBP 1749, and perform studies in Miapaka2 xenografts showing that our drug, similar to the studies reported by McMahon, that it cooperates with trametinib to significantly reduce the growth of tumors in the xenograft. Now, one of the main hot topics in cancer, of course, is immuno-oncology, and there was a Nature paper that appeared back in April from the group based in New York University. And this group demonstrated that, in fact, it's possible to take both genetically inhibit autophagy or use chloroquine to inhibit autophagy and show cooperation of the degradation of MHC1 in pancreatic cancer. So this is basically shows that we can combine this proof of concept from this group, the group of Alec Kimmelman, showing that the combination of autophagy inhibitors with immune checkpoint blockade in PDAC and other chemotypes actually potentially works. So these data are hot off the press, and in fact, we have shown now in cells, and this is from the lab of Ruben Shaw, that our compounds, both the early probe 6965, another more recent probe 7455, and our potent lead candidate, all do the same in cells as chloroquine, but more effectively. And so this is a really key result demonstrating that our drugs will potentially cooperate with immuno checkpoint blockade. And then in terms of the AIM3, we performed a lot of initial IND-enabling studies on our lead candidate. For example, we were able to do kinetic solubility and demonstrated that the compound is soluble in what's called fasted state simulated gastric fluid. And so this is important to demonstrate that the compound will dissolve in vivo in that setting. We also showed a definitive test for potential cardiovascular side effects. So this is with electrophysiology, looking at HERG current blockade in cells. And the results show that a lead candidate does not significantly inhibit HERG currents up to 100 micromolar. So this is an excellent result. We also looked at plasma protein binding across species and showed that for mouse, rat, dog, and human plasma, the compound is highly plasma protein bound. We've also started to profile cytochrome P450 interactions. And so looking at straightforward in vitro inhibition of key P450s, for CYP3A4 and 1A2, we're at micromolar levels in vitro. For CYP2C9 in vitro, we do have a more potent inhibitory activity for the compound. Similarly, looking at CYP450 induction, and again, this is what was detected in this assay, is that there is a low level of CYP induction compound, which potentially meets FTA criteria, but there's certainly something that should be monitored. In another study, we looked at the potential time-dependent inhibition of the compound against a panel of CYP450s, and there were no effects on most of the CYPs except for CYP3A4, and the compound did show time-dependent inhibition of CYP3A4. We also looked at stability in hepatocytes across multiple species, mouse, rat, dog, monkey, and human, and found that, interestingly, the drug has excellent hepatocyte stability in human. Next is mouse, and then rat and dog. So this bodes well because it means that mouse metabolism is at least a reasonable indicator of what the metabolism is likely to look like in humans. We also looked at metabolite ID across in hepatocytes, and again, mouse, rat, dog, monkey, and human. So this was a pretty big study looking at both metabolites of 4 hours and also 24 hours. And basically, several metabolites are formed across a species. And interestingly, glucuronides are formed, suggesting that there's less first-pass metabolism and more secondary metabolism of the drug. Finally, I'd previously shown the mouse in vivo pharmacokinetic profile, orally and IV. We ran the PO and IV arms in rats, and the compound shows very good pharmacokinetics, both IV and also PO, with oral bioavailability of around 70% in rats with reasonable plasma exposure over time. So finally, so this is a Gantt chart just showing where we are in terms of moving the drug through these early IND-enabling studies. The next steps are to perform some other key assays, for example, looking at blood to plasma ratio. We're planning to do a mouse 5-day pharmacokinetic time course study, and also a non-human primate IV PK study and a dog IV and PO PK study, which we hope to get completed within the next couple of months. The only potential issue being the issues related to COVID-19. So in summary, our progress so far in the first year of funding, we've completed 100-gram preparation of the lead candidate ready for non-GLP studies. And we're currently in the process of starting to get quotes to look at a GMP manufacture of more than a kilo of the material. I showed that the hot off the press biological data, our ULK1 inhibitors stabilize MHC class 1 levels in myopathy 2 PDAC cells. And in terms of the ADME and PK studies, the compound is soluble in gastric fluid, highly plasma protein bound, but does not have any cardiovascular liabilities. The compound inhibits some of the CYPs, especially CYP2C9 in vitro, and it demonstrates time-dependent inhibition of CYP3A4 in vitro. So this is, although this profile is actually very typical of this class of kinase inhibitors, and it's important to know this information with respect to future drug-drug interactions and planning for clinical studies. And then again, with respect to hepatocytes, we have good stability in human hepatocytes, and we've noted a range of metabolites that are formed. And then finally, with respect to rat PK, the compound looks really quite promising. And so IND-enabling studies are continuing on with our lead candidate. In addition, we have an active lead optimization process for backup candidates. But finally, I'd like to acknowledge our co-workers in my lab and Ruben Shaw's lab over at the Salk Institute. And I would like to acknowledge and thank funding sources, including our Cancer Center grant, the Epstein Family Foundation, and of course, Pancreatic Cancer Action Network. Thank you.